Alex Bajcz from University of Maine, Orono, and he'll be having a talk not out of the woods. Go ahead.
Just make sure the camera works. Yes. Okay, good. All right. Good morning, everyone. My name is Alex Bajcz, and I'm a PhD candidate at the University of Maine, Orono. Today I'm going to tell you a little bit about my thesis field research, which addresses the question, why does the number of flowers you start off with matter? This question interests me because many plants, both wild and cultivated, produce an abundance of flowers, but produce relatively few fruits. In other words, fruit set is often low. In fact, several people have proposed that low fruit set is probably the rule for plants that aren't good selfers. Now, fruit set is low probably because individual flowers can fail for an awful lot of reasons. These include exogenous reasons like florivery or frost damage, as well as endogenous reasons like genetic defects or incompatible pollen. No doubt plants have evolved to produce many flowers, at least partially to hedge their bets against all of these factors. Now, of these factors leading to flower failure, two tend to garner the most attention: pollen limitation and resource limitation. We tend to assume that if there were enough pollen and enough resources, most flowers would be able to set fruits. But for many systems, this doesn't necessarily appear to be true, including for Maine wild blueberry, which is my system. For those of you who are not familiar, in Maine, fields of wild vaccinium and gustifolium clones are managed like farms. And in keeping with this, they are often fertilized, irrigated, and sprayed to control for weeds and pests. Moreover, last year alone, Maine growers rented something like 80,000 hives of honeybees to assist with pollination. Yet despite this seeming abundance of resources and pollen, fruit set is still only typically 30 to 50%. So we might ask, why do so many flowers fail? Are some of them just doomed from the outset? To try to figure this out in 2013, I went out and removed 60% approximately of the inflorescences from every stem in 30 plots, and paired this with 30 plots in which I counted and handled inflorescences but didn't remove any. Now, I demonstrated many significant changes to growth and development as a result of this flower removal treatment, including to relative fruit production rate, which is what this graph will show. On the y-axis is the average total number of fruits produced per stem at harvest time, and you will note that unthinned plots produced on average approximately 46% more fruits per stem than thin plots did. So thinning did not increase total fruit production by any means. But we might ask if this 46% drop is as much of a drop as we would have expected, given the initial 60% handicap that we put on these plots. To answer that question, I essentially mathematically thinned unthinned plots by that same relative proportion as thin plots were actually thinned by, so about 61%. Now these two plots should produce relatively the same number of fruits, but they notably don't. Thin plots produce on average about 15% more fruits per stem than this projected production amount, which is a highly significant difference, and suggests that thin plots were consistently exceeding their expected reproductive output. Now, this result in particular is interesting to me from a theoretical standpoint. Let's imagine we have a plant with 20 flowers, from which it will eventually produce 8 fruits. 12 of these flowers are doomed to fail, right? If we were to randomly remove half of this plant's flowers, we might now assume that we've removed roughly half of the flowers that previously would have made fruits, and now this plant will only produce 4 fruits. If it instead were to produce 7 fruits, it would seem this plant has beaten the odds. That would mean by chance alone, 9 of the flowers we removed happen to be ones that were doomed. Because I consistently observed my thin plots beating the odds in this way, I can conclude that most blueberry flowers that fail aren't doomed to fail. They fail for some reason or reasons that flower removal helps alleviate. So this realization took me to the literature. I wanted to know if flower removal could stimulate similar changes in other species, and if so, what kind of problems is it fixing there? So I reviewed like 75 recent studies on flower removal and condensed their collective logic into 9 plausible hypotheses for what flower removal might be doing on a mechanistic level. This exercise made me aware of 2 major problems with the current flower removal literature. Oftentimes these mechanisms are just assumed to be operating rather than explicitly tested. And second, when they are tested, they are usually tested in isolation, as though they are somehow mutually exclusive, which I doubt. So I wanted to simultaneously test 3 of these hypotheses in the same system using blueberry as a model. And for that purpose, I chose the 3 resource-driven hypotheses here, which are the ones that I'm going to describe in more detail. The resource limitation hypothesis is by far the most common one you'll see in the literature, and it's also the most straightforward. Essentially, if resources are crucially limiting, then reducing the number of sinks competing for those resources should guarantee all other sinks a greater share of the remaining resources. The spatiotemporal limitations hypothesis argues that reproductive sinks that develop early or otherwise dominant can become such strong sinks for resources that they take more than they should or even need to, and as a result that impedes growth and development in all other sinks. Targeted removal of these dominant or bully sinks, as I like to call them, will allow all other sinks to have better access to resources. For example, in many plants, dominance is positional, with bottom sinks subordinate to top sinks or vice versa. Lastly, the compound interest effect hypothesis states that early reproductive development can sometimes come at the expense of early vegetative development, which can limit leaf growth to such an extent that less total photosynthate is produced over the course of the year. Thus, removal of some reproductive sinks early on can pay double dividends. Not only do you have better leaf growth, which results in more carbon assimilated, but that carbon is being split over fewer sinks. To test all of these ideas, I established 242 plots of blueberry. Each of these plots either received flower removal or not, and the remaining subset received one of 3 additional treatments, each one designed to test one of the 3 hypotheses I just described. So these 3 treatments were repeated foliar nitrogen fertilization, mid-season near-total defoliation, and flower removal that was biased by the sink's position on the stem. Now, if these hypotheses have merit, we should be able to make the following predictions. First, foliar fertilization should help unthinned plots more because they should more desperately need the resource boost. Similarly, defoliation should hurt unthinned plots more because they should more desperately need the leaves that they are able to make. Lastly, biased removal that targets the dominant or bully sinks should have a much greater impact than removal that targets the subordinate sinks. Because we don't know which sinks might be which in blueberry, I had plots in which top sinks were removed and ones in which bottom sinks were removed. So these arrows could easily be reversed for all we know. Essentially what I was looking for is significant interactions between flower removal and these treatments. Before I get to my results, I just want to show you the types of data that I've been collecting for this work. I know this is a large list and might seem a little unfocused, but I wanted to be able to track the physiological changes that were occurring at as many levels as I could. I'm going to return to these purple traits in a moment when I get to my results. I also just want to point out that some of this work is still ongoing, although we're getting closer now to being done. Okay, I just want to hit some of the highlights of what I've found so far. First, I was once again able to demonstrate many significant changes as a result of flower removal, some of which are listed along the left-hand side here. One of these was to fruit ripening rate. This y-axis shows the proportion of fruits that were ripe when I harvested in early August last year. That's about a week ahead of when commercial harvesting started in Maine last year. So we were pretty early at this point. You'll notice that a significantly greater proportion of fruits were ripe in thin plots at this time, suggesting at least that fruits in these plots were ripening at a faster rate. However, unfortunately, the proposed interactive effects that we were expecting to see between flower removal and these other 3 treatments were either rare, ran counter to expectations, or were difficult to interpret for other reasons. As an example of the first of these, I've so far only demonstrated 2 significant interactions between defoliation and flower removal, both of which were related to relative fruit production again, which is now graphed on the y-axis as relative fruits per stem at harvest. In other words, thin plots have already been scaled down in the same ways before. Once again, thin plots do significantly exceed their expected reproductive potential, and in keeping with the compound interest effect hypothesis, unthinned plots are considerably hurt more by defoliation than thin plots are, which actually show relatively little impact of defoliation. Now, while these results are encouraging, they beg the question, if compound interest is a major player in blueberry, then why are its impacts so limited in scope to just these few traits? And I unfortunately don't have an answer to that yet. Meanwhile, I have demonstrated many significant interactions between fertilization and flower removal. The only problem is they're all the opposite of what I would have expected. For example, here we have ripe fruit set, which is the proportion of ripe fruits produced relative to an estimate of initial flower number. You can see that for this trait, fertilization has very little impact on the plots, and thinning doesn't have much impact by itself but plots that were thinned and fertilized do considerably better than every other plot type, even though they really shouldn't have needed the resource boost they were getting nearly as badly as unthinned plots. Similarly, the biased removal treatment also produced some murky results. I was able to consistently demonstrate effects of biased thinning versus unbiased thinning. In every one of these cases, biased plots do better than unbiased ones do, but neither biased plot significantly does better than the other, as you might have expected if one was mostly removing dominant sinks and the other one was mostly removing subordinate sinks. As an example of this, this shows the proportion of inflorescences that remain successfully and actively reproductive the entire growing season. It is a proportion, but it occasionally goes over 1 because it's based on subsampling, but the same protocol is used to quantify it for every plot type. What I hope you'll notice here is that thinned plots of all kinds do consistently better than unthinned plots, and biased plots do consistently better still than unbiased plots do. But neither biased plot significantly outperforms the other. This is a strange result that I want to try to explain, so I have 2 theories for why this might be happening. First, it is possible that in fact it is middle position sinks that are dominant in blueberry, which while rare for plants in general has actually been proposed for blueberry before. This fits with the data because my protocol would have essentially always been removing these middle position sinks. Alternatively, it could be that leaving reproductive sinks in a clump somehow streamlines allocation of resources to those sinks in a good way, sort of regardless of where that clump is left, top versus bottom. I have anecdotal evidence for this as well, but I feel like this explanation is really lacking a crucial mechanistic basis right now. Okay, so I would offer the following tentative conclusions so far from my work. First, flower removal definitely does elicit significant growth and development changes in blueberry, but it's still unclear how it does so. Second, if there is a consistently limiting nutrient in blueberry, it must not always be nitrogen. Third, photosynthate limitations may importantly limit fruit production potential in blueberry, which is definitely something that growers should take note of. And lastly, of the hypotheses I've tested so far, it seems that the spatiotemporal limitations hypothesis is the best poised to explain what's going on with flower removal, but it's also clear that it's going to need further testing and refinement to work for blueberry specifically. Some further questions I'd like to see answered. First, does year matter? Last year was an exceptionally good year, and I'm the only one hoping it, but I hope this year is worse, because I'd like some contrast. I'm doing this experiment again to try to figure out the answer to this question. Second, what is that limiting nutrient if there is one? Is it phosphorus, is it water, or is it a micronutrient like boron? We really don't know. Third, are the effects of flower thinning linear? I've always thinned by the same amount up till now, but I have many plots this year in which I'm thinning by much smaller proportions to try to figure that out. And then lastly, is there a way we could maybe boost early leaf development in blueberry without flower removal? Because if we can, farmers might be much more interested in doing that than removing flowers. Okay, I'd like to thank you all for listening this morning, and I'd like to thank all the wonderful people who have and continue to make this project possible for me. And of course, I'd be happy to take any questions and comments you might have, and I've also provided my email address for you. Yes?
I was wondering, on your plots, are they periodically burned like every 2 years, or are they mowed? And would that have any influence?
So virtually all Maine wild blueberry is pruned on a 2-year cropping cycle. In other words, after it fruits, it's cut down to the ground, allowed to regrow for a year. It sets floral buds, and then those buds burst, and flowers and fruits are produced the following year. Every blueberry field in Maine, basically, that's cultivated is treated like this. At the particular farm I'm doing my work, is mowed in the fall and then burned in the spring. So actually both of them are done. And that's to remove litter for pests. So it's hard to know because that's virtually the condition in which all Maine wild blueberries are growing in when we're talking about cultivated. So, yes.
Do you see any problems with fungal issues on flowers or stems?
I haven't noticed them a lot in my plots, partly because we do spray for things like monolinea, which is one of the main fungal pathogens. There are others. Certainly floral blights are one of the main reasons that flowers fail in blueberry, but it's probably not even in the top 5, surprisingly. There are a lot of other reasons. I think maybe chief among them is self-pollination, which is a very big problem in blueberry.
Alex, I'm wondering when you do these sort of flower removals, do you see any difference in pollinator behavior, either because of the difference in inflorescences or because of secondary compounds that are getting released because of the removals?
Yeah, so I didn't address it because I didn't want to dwell on it, but if we return to this slide, you'll notice that these three in the middle here are all pollen-driven hypotheses. I emphasized earlier that pollen and resources seem to be where most people think the problems lie, and I didn't test them, but I'm certain that there could be something here in terms of significant changes to
Well, I was just
pollinator
wondering
behavior.
anecdotally if you had noticed anything.
Well, I wouldn't be surprised because, for one thing, thin plot fruits are much bigger. They're maybe about 15 to 20% bigger in some cases, so a significant difference. And then blueberry fruit size is super strongly correlated to the number of mature seeds you've got in there. So it seemed like very unlikely to me that there was no impact of pollination as a result of flower removal. I just haven't been able to demonstrate it yet, and I hopefully will be able to soon, because one of the things that we're currently working on is seed set. I just couldn't find a way to sort of streamline the process. It was taking about 20 minutes per fruit, and we don't have that kind of time, so I'm trying to streamline this, but we're getting started, and so hopefully I'll be able to at least speculate on that.